Hello. Um, Today I think I'm gonna mainly talk about sequencing. So we know what DNA is. Many people have heard of the word called DNA. Um, so DNA has is made of bases, like there are four letters, um, A, G, C, T, and in any combination of AGCT, the DNA is written. So, like, maybe something might go like ACCCGTTTCCAAAGTC and goes on. And if there's one mutation in one of those bases, it might affect the genetic expression or other biological issues. So people, scientists, very brilliant scientists, developed this technology, the method, which helps you to sequence them. So sequence, so in this case, sequence means you read each basis. So does C comes after A, or like A comes two stops before C, like how what how do they look like? It's like alphabet, so like you basically um write out alphabet from A to Z. It's same thing. So like from here to there, what's the combination of how A, C, T, G are situated or located. So far, and recently, how people sequence them, so how people read the spaces or short reads. So they only read 100 letters at a time, and they kind of read from first position till 100 position, and maybe from 10 position to 110 positions. So they read short pieces of letters, and they and then because there are gonna be some overlapped region, so based on those regions they kind of speculate the whole sequence from it. And as methods got developed more and more, people that method became very accurate. So they do have like almost 99% accuracy to read sequence those bases. However, because those only reads like fragments, like small fragments of DNA or RNA or anything else, it got difficult to see the big picture of the region. So for example, one gene runs from zero, like position 1 till like say 5,000 position. Then if you read like small bits and pieces, like many of those, and then make the whole get the whole sequence, you won't really know how much of how many small mutations there are, or there's gonna be like some discrepancies between the actual gene sequence compared to what you sequence from the with the short read sequences. And there are some things called splicing pattern, like ultimate splicing patterns. So there are some times that like some positions are missing and some positions are like added, or they're combined to each other. So if you see like small pictures, many of small pictures, and then speculate from that, it became very accurate to guess which letters comes where and which letter comes where, but you can't really tell the big picture of that region. So people made this method called long read sequencing, which reads the sequence, like those all bases, in one go. So they read up to 10K, which is like 10,000 bases at one time, and they can guess, or tell, it can tell what base comes where. So all the like sequence at one go, and because they can do it like the big chunk at a time, they can tell us like quite big picture of that region that we're sequencing. Like there are these regions like combined to each other, or some region is empty, like omitted, like so there are many different events going on within that region. So that allows us to like know and learn more information than just what bases there are, like what that region has, like sequences only. So yeah, that's like good things about it, because but at the same time, because it's not like it just reads one time the whole big chunk of fragment, it has quite low accuracy, because if you read small fragments over and over, then it kind of gives more confirmed sequence, whereas if you read big chunk at a time, it's like, yeah, that it's read okay, but still not 100% confirmed by with the overlapped sequences. So it kind of has lower accuracy, but when we say lower, it's not like bad in an accuracy way. It's like lower than short read sequencing, but still very good. So nowadays people use, like many scientists use this technique to investigate different forms of transcripts. So one gene might have several different transcripts that might have different functions in the end, and to investigate that, people use a lot of long reads sequencing, and also if they want to study like environments around certain regions, like methylation levels, or what else do they do? They even can do single cell sequencing with long read sequencing, I think, right now. But to see the environment around that region, they also use long read sequencing. So nowadays many people apply this technique to their own research, and give us very good knowledge, insights in terms of biology, like genetic insights. So I use this technique to investigate a gene that is studied to be risk gene for psychiatric disorders like schizophrenia or bipolar disorder. And also I personally studied these two genes that were known not to be expressed in the human brain, but the short fragment of these two genes have expression in human brain. So I am trying to look into what those are and how they look like, because this is like novel finding. I still need to know how they look like the sequence of them, and then whether they are aligned to those two genes that I think they are aligned to, or just like randomly like read random different gene to know that I used sequencing technique. Yeah, and or my friend uses this technique to compare one risk gene for schizophrenia disorder in a human brain and in human cardiovascular system, or mice brain and mice heart, so that if two genes have different sequences or different types of transcripts in each tissues, it gives like really important insights when we develop medicine, because you'd like to, in the end, you would like to like develop a medicine that only targets human brain rather than heart, to cure schizophrenia or bipolar disorders or any depressions. So yeah, so today I talked about sequencing, and I hope it makes sense. I tried to say it very like easy way, but I couldn't really think of any examples that's easy to understand. But yeah, so next time I'll come back with more interesting knowledge, I guess. Bye bye.